So good morning, ladies and gentlemen. I think we need to start because we're also live broadcast. So a very warm welcome also on behalf of Bob Schön from the United States. A very warm welcome to the session that is dealing with paradigm shifts in rheumatic diseases. And I'm very pleased to deliver a lecture on the wind of change in rheumatoid arthritis towards targeted synthetic and biosimilar DMARDs. And so this title was assigned to me by the scientific committee, and I'm very grateful to the scientific committee. But when you know that you are assigned such a talk, you know that you will lose days of work on that talk, and it may not be perfect in the end anyway. But I tried my best to encompass these two areas of new targeted synthetic DMARDs and also the biosimilar DMARDs. And these are my disclosures. And I would like to start. Why Wind of Change? Some of you may know that this is a very famous song by a German rock band called the Scorpions. Actually, they are from my hometown, Hanover. And this song was written, Wind of Change, in 1989, 30 years ago, right after the wall had come down in Germany, as you see on the right-hand side, and also led to the invitation of this rock band to Michael Gorbachev in 90 to his place and to report about the wind of change that had occurred in Europe during that time. And that was quite impressive and remarkable, and because we have been living in peace for more than 30 years after the wall had come down. And I think it is very, very important to stress the European spirit after this event. So let's go now to rheumatoid arthritis and 25 years of biologic therapy in rheumatology. What have we achieved and where are we going? So we can usually we can avoid damage usually completely with immediate therapy and application of the treat to target principle. Nevertheless, usually only a maximum of 50% of our patients achieve the complete remission and complete discontinuation of medication unless you treat very, very early, and maintenance of remission is rarely possible in seropositive patients. Therefore, there is still a high demand for new therapeutic principles. And this slide shows you the timeline of the development of anti-rheumatic drugs, particularly with respect to rheumatoid arthritis. I think we have basically witnessed 4 milestones: glucocorticoids, methotrexate, biologics, and then the new small molecules. And you may also include the biosimilars in this area. So we started with the biologics roughly around the millennium change. Then the small molecules came somewhat later, and around a couple of years ago we had the biosimilars, and there's still new drugs in development that you can see here in the small molecule Jak area. So if we now look in where do these drugs fit into the current management recommendations, and I took this out of Professor Smolin's paper from 2016, and I would like to remind you that tomorrow at 9 o'clock the new updated management recommendations will be shown. They are not that much different from this particular part here, so I used it. And so if you have failed conventional synthetic DMARDs plus glucocorticoids, then you move to phase 2. And if there are prognostically unfavorable factors present, then you add a biologic DMARD or a Jak inhibitor. And the biologic DMARDs, as we already wrote down in 2016, can either be the original biologic or a biosimilar biologic. Also, we have now the targeted synthetic DMARDs, which are primarily the Jak inhibitor. Currently available and approved in many, many countries are Tofacitinib and Borreliosis. So what are the advantages and disadvantages of biologics? The pros are they're very specific. A monoclonal antibody against TNF only binds TNF and nothing else. So therefore you have few off-target effects. They might relate to injection site reactions or allergies sometimes. They very often have a very good safety profile. They have a long half-life, which may be very important, just one injection per month, for instance. May also be a disadvantage because you want to get rid of a drug in case a problem arises. And you have, from a company's perspective, from the development perspective, they have a good patent situation and also a good revenue situation. And the biologics, as you have seen, have been the most sold drug ever on Earth. The cons are, it's a complex mode of action because you interfere with a certain cascade. Sometimes there is a difficult dose finding. They need parenteral administration. This is what our patients often ask us. They are quite expensive to manufacture because they're not small molecules. They are sometimes complicated protein structures, and there may be antibody formation against the therapeutic reagent, the so-called anti-drug antibodies. So therefore the question arose, can we overcome some of these issues with new oral small molecules? And so that's why we call them sometimes honorary biologics, because of obviously they are not protein structures, they are small molecules. But it was quite interesting when I reviewed the several hundred abstracts submitted in the Biologics section. Quite a few Jak abstracts had crept in, because apparently the authors thought these were also biologic, but of course they are not. We can only call them honorary biologics. The question is, by using them, can we say time to say goodbye to syringes, as you see here on the upper panel? I personally absolutely do not believe so. I think the biologics will stay and are very important, but it's very nice to have additional medication as well. So let's start with Jak inhibition. And when I asked my student and my fellow rheumatologists, what does Jak stand for? And they say, oh, Janus Kinase. But indeed, many people do not know that when they were initially detected in the group by Andrew Wilkes, they were just called just another kinase, Jak. And why so? Because when they were analyzing certain enzymes in hematology, they come up with some bands of proteins which were enzymes, and they didn't actually know what they meant, and they were not really interested initially in these enzymes, and so they said, oh, just another kinase, and they called them Jak. But then it became apparent that the JAKs are very, very important molecules, and if you want to submit a paper like you see down there, the journal editor will say, what? Just another kinase? So we have to rename them, and that's what they did. They called them Janus Kinases, and the paper in the upper panel is highly readable. I highly recommend to read it to you because it's written in a very good way. You can read it at night in your bedroom because it's so nicely written. So this is Janus Kinases, and now the official version is Janus was the Roman god looking into the old year and into the new year. That is because there are two domains in the Janus Kinases that are displayed here, and that's why the name arose from. But as I mentioned, it was originally a different thing. And also the development of these Jak inhibitors is quite interesting, because it started, to my knowledge, with a scientific conference in 1993. So quite a while ago, researchers got together and people from industry got together, and they were looking into signal transduction mechanisms in inflammation, in autoimmunity. And then finally, Paul Jangelian from Pfizer and one of the heroes in immunology and signal transduction, John O'Shea from the NIH, got together, and they thought, oh, it might be an interesting target, and that's how it started. And finally, a drug was developed that this was then followed up in research and paved the way for this modern development. In parallel, and a little later, of course, other Jak inhibitors were developed, such as Baricitinib that you see here. And frequently there is a startup company that generates the first reagents, generates the idea, a larger company steps in, because you see the very, very extensive program that is currently needed in order to have a drug approved subsequently, and that is shown here in the Baricitinib timeline. So how do they work? And I now could show you all kinds of diagrams with fancy arrows and enzymes and so on, but I will make it simple. So I took this picture from Amsterdam, which you can freely download from the website. And this is actually a bicycle. And you can only ride the bicycle well if you have a saddle. But if the saddle is blocked by a molecule, it's very difficult to ride the bicycle. And if you want to ride, but the bicycle drives the cart of inflammation that you see on the right-hand side. So how can you block the saddle of this bike? Well, you can put on these little ducts, and these could be either Tofacitinib, Baricitinib, Upadacitinib, Filgotinib. They sit on the saddle. Then there is no signal transduction anymore. You cannot use your legs in order to keep the bike moving, and so therefore the cart with inflammation is not moving as well. In the background, you see a little animal, which is the TNF inhibitor, and you see that this animal has nothing to do with this bicycle. It is a completely separate way of signal transduction and signal mediation apart from the JAK-STAT system. But of course, I need to show you this as well. So the JAK system is composed of basically 4 chains, JAK1, 2, 3, and TYK2. And they're quite promiscuous. So they marry, one chain marries to another chain, and this chain marries to itself, and so on. So that's why you have the composition of JAK1 and JAK2 as one molecule, and they're all receptors for very important cytokines and interference. So if you interfere then with a certain JAK inhibitor, then you also interfere with the action of these cytokines. They can no longer work, or at least they are reduced in their action. And in order to block these various chains, you have these new reagents like Tofacitinib, Baricitinib, Filgotinib, and Upadacitinib, and also Tofacitinib, and some of them block several chains of the JAK system, such as Tofa 1, 2, and 3. Some JAK1 and 2, like Baricitinib, and some are selective for JAK1, such as Filgotinib and Upadacitinib. Obviously, they're all small molecule. That's why you can give them as oral medication. How do they work? And now I have to show you some biochemistry. Don't worry, it's not too much. But you see on the left-hand side, this is the currency of energy. This is the ATP molecule. And many of you still might remember your early days of biochemistry in medical school, that this was a very important molecule being responsible for the energy metabolism in the body. And you see the similarity on the right-hand side with the JAK inhibitor. And if you now go to another model, it's like a vending machine. You see that on the right-hand side, you see a 2 euro coin and a Thai coin from Thailand. And many German tourists took these coins along when they went on vacation to Thailand, because they worked in the vending machine as the same as a 2 euro coin. But then, of course, the vending machine manufacturers detected this, so you could still put in the Thai coin, but the machine didn't work. While the machine would work if you use the 2 euro coin, the ATP. So the machine is blocked because of that coin, because it's a wrong one, but it just abolishes the action of the vending machine. And that's the model how these JAK inhibitors work. So how do you assess JAK-STAT selectivity? And I would like to address and focus your attention to a very nice article written by Ernest Choi just recently on JAK-STAT selectivity. We have to go a little bit back to biochemistry. So if you need a lot of material to block an enzyme, the selectivity is very low. If you need just a tiny bit, then the selectivity is high. And then you have on the left-hand side the various compounds, and you see that they all have different requirements for the special drugs, and therefore they mediate, in this case, the selectivity as you see here. You can also look into certain animal experiments and animal models, and you see here the interesting display of the action of tofacitinib and upadacitinib with a different selectivity for these chain, and you also see that there is a different selectivity of tofacitinib and in this case upadacitinib, as shown here in this in vitro model. How this refers to clinical action and to the side effect profile, we don't really currently know entirely. So that is very important on our research agenda. So let's start with tofacitinib, and I will not go into many of these details because this drug has been around, and this is a win session for quite some time, but it's very nice that just recently this paper came out by Jürgen Wollenhaupt and colleagues assessing the safety and efficacy of tofacitinib for up to 9.5 years. So it's close to 10 years that we have clinical trial data, and that is very reassuring for our patients because they ask, doctor, how long has this drug been around? Is this a newcomer? Is it really safe? And that was addressed here. I will just show you the efficacy data. It's always a little bit difficult to assess long-term studies because patients not tolerating the drug or not being effectively treated drop out. So therefore, it is always with a grain of salt. You have to assess these, but you see that it is very stable over nearly 10 years of usage. So that, of course, led to the development of additional drugs. Some drugs died because there was some complicated pharmacology and pharmacokinetics to it, like disorinutinib, even though it showed good clinical data, but tofacitinib, baricitinib survived, and others are in development. I will come to later. In this very same paper, obviously you cannot see it here, but they did meticulous work on showing the most recent phase 3 data, but also the phase 2 data on the right-hand side. So now let's go to the second drug, which is called baricitinib, and that drug has also been investigated in several settings. Let's start with the, let's say, normal setting that most of the drugs are initially investigated, namely in those patients who do not respond sufficiently to methotrexate in this case, or to other conventional synthetic DMARDs. And here baricitinib was put on top either 2 or 4 milligrams per day on top of methotrexate, or in this case patients received in addition placebo or the active drugs, and you see in the lower panel that it was quite effective in this patient population, and also the safety profile was acceptable. RA Begin was a study that started with patients who had been naive. That is the second group that we are interested in, even though this is a rare clinical situation, because normally, according to the recommendations, we start with methotrexate or a conventional synthetic DMARD. Nevertheless, it is important how does the drug fare, and you see also that baricitinib in these 2 dosages was superior, in this case, to methotrexate. RA Beam addressed the question, what about patients who have also failed methotrexate, but are in parallel treated with what we called our golden standard of treatment, adalimumab. So here in the RA Beam trial, which was a very important trial in the history of trials in rheumatoid arthritis in general, not only compared placebo and then switching to bari, but baricitinib with adalimumab in an initially non-inferiority design with the possibility to go into the superiority assessment. And what you can see in the lower panel, that the drug in 4 milligrams per day was superior to adalimumab. So that was the first sign that you can generate additional efficacy with such a drug. RA Beacon addressed the question if the drug also works in patients who have failed biologics, and as you see here on the lower panel, yes, indeed, that was possible. So what about the newcomers? So Tofacitinib is a new drug that has been developed, and you see here that there was also an extensive program. You see that, you know, the name. Oral was always oral stands for TOFA trials, RA for BARI, and SELECT is the acronym of the Tofacitinib trials. And you see the drug was investigated in methotrexate naive patients, inadequate responder patients to methotrexate, also in patients who had failed biologics, and also will be compared to, in addition to another drug, as you see here, but that trial is not finished yet. So SELECT next is usually the first starting point in a trial in phase 3. So here we used a placebo. Of course, all patients were still on methotrexate versus Tofacitinib, either 15 or 30 milligram a day. And you see here, compared to placebo, the superior efficacy of that drug. What then was quite interesting to us, if you looked into a population that had failed sometimes many biologics even, and did the same analysis in these patients who had been biologic inadequate responders, you also see that the efficacy was quite good, not actually much different from those who had failed only, in quotation mark, methotrexate, also quite reassuring in this aspect. And here we have the group around Ronald van Vollenhoven looked in patients who had been methotrexate naive. They also received Tofacitinib 15 or 30, and it was shown that there was a significant and clinically meaningful improvement compared to methotrexate, and also radiographic progression was significantly less with the Tofacitinib compared to methotrexate. And the next study addressed, again, like we saw with RA-Beam, the question, what about the comparison to adalimumab? And this was done here by Professor Fleischmann and colleagues. In this case, Tofacitinib 15 milligrams per day showed superior on improvement in RA signs in this methotrexate radiographic population, also towards not only to methotrexate, but also to adalimumab, as you can see here. On this curve, Tofacitinib is the green column, which is also superior to adalimumab in this particular study. So what about the overall analysis of JAK inhibitors versus this TNF inhibitor? And I've compiled this on this table here. I alluded to the study RA-Beam by Peter Taylor, method Baricitinib versus adalimumab, and the superiority analysis was met. When we looked into Tofacitinib plus methotrexate versus adalimumab, a similar finding was obtained. In Tofacitinib plus methotrexate, adalimumab plus methotrexate, the oral strategy showed that there was a non-inferiority finding. It was not met when you compared Tofacitinib mono versus adalimumab plus methotrexate. And the non-inferiority study that is shown here at this conference, called FINCH 1 with filgotinib, showed also the finding of the fact that non-inferiority was met. So here are the data of the oral strategy, which was also a milestone in the development of JAK inhibitors. So let's have a look at filgotinib, which is quite prominently present here at this meeting as well. You see here the overall, they are all called FINCH. So if you ever see FINCH, it's filgotinib. And FINCH 1 addressed methotrexate with the active comparator adalimumab, and FINCH 2 addressed conventional synthetic DMARDs, and then FINCH 3 methotrexate, and I will show them. So here this was recently shown at the ACR meeting by Mark Genovese, that in this phase 3 study with highly active rheumatoid arthritis and prior inadequate response to biologic DMARDs, the treatment with filgotinib was successful, and you see here the data on display. And also the DES28, so this was the ACR20, also showed superiority. So what are the additional findings about filgotinib? I will not go into the details because they are here on display or have already been shown. These are the Finch trials that you see here, and please have a look at these contributions here at Eular. So finally, it's some words to Tofacitinib, which has recently approved in Japan, I think just a couple of weeks ago. And also there is an extensive phase 3 program, and the data were presented at ACR by Dr. Takeuchi, and could also show inadequate patients who had an inadequate response to methotrexate. Tofacitinib, they tested this here in 100 milligram, 100 and 150 milligrams. That was also superior to the placebo and showed also an acceptable safety profile. And you see the data here with the two dosages compared to placebo. They also had in another trial, they had also an etanercept arm, but that was an open arm, so you cannot really compare this like an RA beam or in select compare, because patients knew that they were receiving a biologic with syringes and so on, and that might explain the fact why etanercept here had numerically higher action, because you cannot. Because it was not powered to show this and designed to show this in this particular trial. Of course, what is frequently and always addressed is the safety item, which is of course most important to the patients. And I already showed you the paper by Jürgen Wollenhaupt, so they not only addressed the efficacy items, but I think even more important in such a study are the safety data. So more than 4,000 patients were investigated, and the safety profile remained constant, and for all cause of AEs of special interest, the incident rate, meaning an incident per 100 patient years, were 3.5 for herpes, which is a signal all across the JAK inhibitors, and 2.4 for serious infections, and you see the low numbers below that. Just some examples how stable this was. This was serious infections over time, and non-melanoma skin cancers over time. And finally, you see the herpes zoster signal, which is evident in treating with tofacitinib, but is so also with the other JAK inhibitors. So what about the other drug? This baricitinib also was shown recently in a paper that came out just a couple of weeks ago, also roughly close to 4,000 patients, looking into more than 6,000 patient years of exposures. And over time, there were no differences in the occurrence of these side effects, and no major differences between 2 and 4 milligrams. And herpes zoster was significantly more frequently in the 4 milligram version. And of course, we have discussed quite a bit the deep vein thrombosis, and I will come to that, but there was no difference between 2 and 4 in the overall program, and the incidence rate in this case is roughly what you would expect in the background population. So an even longer period was shown recently at the EPLA conference, and I'll show you these data presented in Brisbane by Stephen Hall. And you see here, I will not go into all of these details. Again, herpes zoster was an issue, and if you look now into deep vein thrombosis, there was a signal in the first placebo-controlled trial, but in the overall trial it was no longer evident. So if you are interested in the safety aspects of these drugs, I would like to refer to you to these posters and presentations that will be have already been shown yesterday, but will be shown today as well. And there you will see detailed information. And please go to these posters to talk to the doctors about the safety profile with the latest news here at Eular. Of course, many of us are aware of the recent alert, both by the FDA and also by EMA, that in one trial, as I show here, 10 milligrams twice a day in Tofacitinib led to the notion that patients should not get twice 10 milligrams in Tofacitinib because there was a higher incidence of deep vein thrombosis and pulmonary embolism. We don't know the exact data because it is still blinded. It was initiated by the Drug Safety Monitoring Board and then related to the authorities, so we do not know the exact data, but this is a clear message that we should not use twice 10 milligrams of Tofacitinib, even though we usually do not use it anyway because the approval is for 5 milligram twice daily. So therefore, it was quite interesting to see the report by Joel Creamer just yesterday with a huge study they performed in the Corona trial, and they could not find a higher incidence of MACEs or serious infectious events compared to biologics, and not in the abstract, but it was shown there was also no apparent signal with twice 5 milligrams for pulmonary embolism. Also, let's have a look at the safety profile of filgotinib. Obviously, we do not have that many data yet, but also this was in line with what we have seen, and also no major signal popped out here in this particular trial. Maybe infection, somewhat more numerically, which is of course not unexpected. So to finishing up the Jak part of my presentation, several small molecules are either approved in many countries like Tofa and Bari, or are in clinical development, largely completed or even with Tofacitinib led to approval in Japan. And what I think is important also as a scientist who's interested in signal transduction mechanism, they support the pathogenic significance of the cytokines involved here. The clinical effectiveness has been demonstrated in all the situations that we are used, and the safety profile does not differ apparently from biologics. The questions still relate to target, Jak target specificity, Jak1 versus Jak2, Jak3, and so on, and the benefit-risk ratio, question about pulmonary embolism, comorbidity, and the herpes zoster signal is clearly there. And now, back from the biologics, we have to think again in terms of classical pharmacology with pharmacokinetics, interaction with cytochromes, and so on, which is important. So to finish up Jak inhibition, this is a picture I took at the most recent ACR conference. So you see the JAKs are distributed there, but if the cytokine comes on, it is a very nice compilation of the JAKs that you see here in this particular picture. So now let's move on to the biosimilars in rheumatology. There's also a wind of change in this particular area, and you see I've compiled some of the names of these drugs here, which are available in many countries. And also, just recently a very nice paper came out by Chris Edwards, and those of you who are interested in that subject, just came out a couple of weeks ago. It really meticulously analyzes the switching to biosimilars and the current perspectives here. I will just show you the example. So which drugs do we have available? It's quite a bit. So we have infliximab biosimilars, we have etanercept biosimilars, adalimumab biosimilars, and rituximab biosimilars. You see the various names here, and there have been, and you will not be able to read it, so that's why I refer you to the paper. Numerous studies have done switching experiments, as you see here in these two tables. And so I like to quote from that paper. Switching patients from reference biologics to biosimilars may lead to significant cost savings. We all know that this is very important. And that can be used to increase the access to biologics within the same budget constraints. So if a country only gives out a certain budget of money for biologics, obviously it's much better for many patients to get the drug rather than for only a few patients. So that is an important statement. Randomized biosimilar development and approval, this is also known to all of us, is a rigorous process, both by the EMA and the FDA and the Japanese and other authorities. And so they need to show the clinical equivalence of the biosimilar and the reference product. And the many trials I just showed you have seen that there is a lack of impact from switching from a reference product to a biosimilar, or with regard to immunogenicity, safety, and efficacy. And also emerging real-world evidence shows this. And of course, there is a lot of discussion in medical societies regarding switching, and ongoing real-world experiences are important here. And just last week, these two papers came out in Arthritis Research and Therapy, and it's very difficult to even discriminate what is the original product and what is the biosimilar if you switch, because the data are basically exactly the same with regard to efficacy in this case. Interestingly here, the German rabbit registry could even show that a biosimilar product had a higher uptake and retention rate compared to the original product, as you see here. And what was the reason? Because the biosimilar had less injection site reactions compared to the original product, and that's why patients prefer to stay on here. So this is also an interesting finding. Of course, there is a lot of debate with regard to interchangeability, and you see the definition on the right-hand side of the EMA, and this is handled differently from country to country or from continent to continent. So here you see how the United States are handling it, how it's handled in Europe, in Canada and Australia, so you have to consult your local authorities how it is done. But very interestingly, just when I prepared my talk, this press release came out from British Columbia in Canada, and I will read it to you because I think it's so interesting, because the Canadian province of British Columbia has said that the public drug plan will switch as many as 20,400 patients from 3 branded biologic products to what they say cheap, I would say less expensive, near copies called biosimilars, saving an estimated nearly 100 million Canadian dollars over the 3 years. And so that, of course, is quite a very interesting news, that you really have to change as a doctor unless there is a certain item that prevents you from doing this. And the reason is, as the British Columbia government said, biosimilar are a necessary step to ensure pharmacare and provides existing coverage for more people and funds new drugs well into the future. So this is a very, very exciting development here in this particular area. Also, the EMA looked into this, and there is a very interesting report by EMA looking into the cost savings, as you should see here. And they give you an estimate how many euros will be saved by using biosimilars. Could be up to 100 billion by 2020 in the 5 countries listed here. And so this will, and they also state that it varies quite a lot across countries in Europe. They have provided this table, which is a number of years old, but it shows you how very differently the usage of biosimilars is handled in the various countries of the EU. Some countries, more than 80% of patients are biosimilars. In some countries, very, very few. And here, what about the American situation? That shows you the cost saving that is projected in this particular paper here, what it might save in cost using biosimilars. So my final take-home message with regard to biosimilars is a significant number of biosimilar biologics have been approved in the EMA region, also by the FDA. Infliximab, etanercept, rituximab, and adalimumab. And biosimilar development and approval truly are rigorous processes requiring demonstration of the biochemical and clinical equivalence of the biosimilar reference product. And I personally believe that switching should involve an intense discussion with the patient and should be ideally be based on a shared decision. But you see, the situation in British Columbia and Canada will be quite different, because there you are forced to switch. So that will be very difficult in that discussion. So very substantial cost savings are possible despite the high cost of biosimilar development. And mind you, it is not so easy that you just go to some garage and with some little beakers you produce a biosimilar. No, this is very costly. It is a rigorous process which can cost up to 300 million euros. And so eventually these costs have to come back to the company. So this is also an important item. And there are different regulations and or incentives using biosimilars in the various EMA countries. You have tenders, you have quotas, and even in Germany we have 16 different ways how biosimilars should be prescribed. In some areas of Germany, a lot of patients are on biosimilars. In some areas of Germany, very few, according to the local regulations. Of course, we also have a number of practical questions regarding the use of biosimilars, such as, very simple, how many drugs can you memorize? Can you memorize all the drugs that you're using? I already talked about the Jak inhibitors, now the original biologics, now the biosimilars come along. Which drug do you choose actually? How much information material can a rheumatologist actually store? Because I get so much information material, I don't know where to put it in order to hand it out to my patients. Obviously also we need to avoid a nocebo effect when a patient realizes, oh, I'm not getting my trusted drug anymore, I get a new stuff. Is it as good as my original drug? May not be as good. So there might be a nocebo effect when switching, and I think that needs to be taken into account as well. Nevertheless, I think I would like to end with a quote again by the Scorpions. So take me to the magic of the moment on a glory night where the children of tomorrow share their dreams with you and me. So we hope that we share dreams, that we have even better drugs, even better combinations, and even better strategies using these newly developed medications. And with these words, I thank you very much for your attention. Thank you all. So we started a little bit late, so that's why we have about 5 minutes of discussion. If you have any questions, please come forward. Yes, microphone number 2, please.
Yeah, thanks for the nice talk. But as far as I know, in the biosimilars are nearly identical to the originator in the matter of efficacy, safety, pharmacokinetics, and even immunogenicity. And you have told us today that the reaction or hypersensitivity to the biosimilars is somewhat less than the originator. I don't know the mechanism for that. Yeah,
I would not say it's a hypersensitivity. I would probably say it's the composition of the stabilizing or the other material that the agent is in. So that's why sometimes it's more an acidic product or so. That's why you have these injection site reaction. It's not a hypersensitivity.
Right.
Yes, microphone number 2, please.
Sumaira, Pakistan. I just want to know, do you do varicella antibodies before starting patients on Jak inhibitors? Is that like a...
Yeah, I can only refer to the German situation. So there are 2 camps in Germany because there's quite an uptake of the new Jak inhibitors, and about 1/3 of patients are directly switched from methotrexate to the Jak inhibitor, and 2/3 get a biologic first. So there are arguments that some colleagues say, well, it is so much easier to use an oral drug. The patients, when being informed what are the possibilities, opt for an oral drug. And some rheumatologists say, well, I mean, what do I do when a Jak inhibitor fails? Can I then go back to a biologic? That is not known. So I will reserve the Jak inhibitors for the more difficult situations. So it is, I think it is very important to discuss it with the patient and tell the patient about the various possibilities, and then decide together with the patient based on the information.
Yeah, I meant, do you do varicella antibodies, like before starting them on Jak inhibitors, Do you test?
Varicella. Oh, I'm sorry.
Yeah,
I did not get this.
No,
I'm sorry. No, what I do is, what we do is we always recommend herpes zoster vaccination. So I do not do the titer.
Okay.
And so I can declare that I was probably among the very first persons in Germany to be vaccinated against herpes zoster. Okay. And I can also add, to my surprise, I didn't experience any side effect or any itching or something on my arm. So, but that I would rather recommend this than doing the antibodies.
Okay.
And then microphone to the right.
Gerd, could you comment on combination therapy? Combination therapy of small molecules with DMARDs is well known in trials and in real life. But combination therapy of small molecules and biologics, there's patients around the world that are doing this, and apparently safety issues are not showing up. Can you comment on this?
I fully agree, and I can tell you, I have approached several companies. Some even produce a JAK inhibitor, also a anti-TNF inhibitor, in order to do these trials, but I have failed. So far, because maybe of the old data, when combining two biologics with higher safety risk, the companies have refrained from doing this. But I think it is very important. I think we need these trials in order to get even better, potentially better response. So,
thank you.
In the way back, microphone, and then.
You spoke very little about the use about new biologic targets for biologic drugs, and you focused more on small molecules. Do these winds of change signal a paradigm shift in which the end of the biologics is near?
They are the end of a paradigm shift at upper management of companies. That is very sad, because I was a victim of that, because we developed a drug together with a company against the GM-CSF receptor with, in my eyes, a very good safety profile, a very good efficacy profile. The program was not followed up. I don't know the reason, but we as investigators suspect that upper management said we will not get enough money back with this compound, so therefore they stopped the development and sold the drug. So that is, I think, a sad development, because I think we need more targets also for biologics, but we need to convince upper management that it's worth investing.
Thanks.
Final question, please.
Hi, I am asking about the maximum dose of methotrexate when combined with tofa or baricitinib to avoid infection, especially herpes zoster, and if herpes vaccination is for all patients or certain group should be vaccinated.
Well, I think, first of all, every patient with rheumatoid arthritis who comes into your office initially should be informed that there is the vaccination possible, and I would do it in my program. I think that is so important, because it's very difficult once you say, oh, I must not treat the patient, but can I do the vaccination? You know that these both shots are 2 months apart. You cannot possibly wait, and so we need to have more efficacy data. Can you do this safely on the background of the JAK inhibitor? These data are currently generated, so we don't know this aspect yet. And the next question was regard to the dosage of methotrexate, and I would probably not go beyond 15. This is our standard dosage, and maybe if we can draw some findings from the biologics, maybe 10 are even enough. Even though we don't know this, it would be interesting to look into the various dosages that can be done with the various trials, what was necessary for a good response. Okay, so I'm afraid we need to move on, and it's another exciting subject we are now covering with Professor Robert Schön from New Haven in the United States. So thank you very much for coming over the ocean and informing us about new and exciting developments in infection-associated arthritis. Thank you very much for coming.
Sir, thank you. I'd like to thank the organizing committee for inviting me to participate in this Congress, and also for the opportunity to spend a few days in this beautiful city. Good.
Let's go out.
Yeah, all right.
You can click there.
Great, thanks. Just click there.
Yep.
Double click maybe.
Double click. coming. It's coming.
Got it. Okay. So I'm going to shift gears. I've been asked to give an update about infectious arthritides, and I've chosen to talk about septic arthritis, Lyme borreliosis, and chikungunya. I'm going to start by talking about septic arthritis, and I think I'll frame the discussion initially in terms of some of the basic principles of management of these patients. But since this is an update, I thought it would be of interest to talk about not so much the specific safety details, which we've just heard about in the last wonderful lecture, but to look more at the landscape of, in a global sense, has the situation with respect to infectious arthritis, septic arthritis, changed generally from the pre-biologic to the biologic era? And then I'm going to change directions and talk about two emerging infections. And you might think Lyme disease, chikungunya, these things are different. One is a bacteria, one is a virus, one is northern temperate, one is more warm climates. But there's a lot of commonalities if you think about Lyme disease and chikungunya. They're both emerging, or probably more accurately, re-emerging infections. They both have different clinical characteristics. And one of the things I want you to take away from this talk is I want you to have a clear feeling of what Lyme arthritis looks like clinically, what chikungunya arthritis looks like, just as you recognize a patient with gout or a patient with rheumatoid arthritis. Patients with chikungunya have specific clinical features. But in the context of my how to treat talk, I want to emphasize the idea that Lyme disease and chikungunya are bimodal infections. Some patients recover completely after the acute phase, but some don't. Lyme disease patients go on to develop Lyme arthritis, which is generally more antibiotic refractory than the early stage of the illness. And patients with chikungunya go on to develop chronic arthritis about half of the time. Why is that? And that's what we'll cover. So let's begin with a discussion of septic arthritis. As you know, the synovium is vulnerable to bacterial seeding because it's vascular and lacks a protective basement membrane. So most septic arthritis results from occult bacteremia following disruption of skin and mucous membranes. We think about septic arthritis as primarily involving gram-positive organisms, particularly staph. Consider, for example, that in one study, gram-negative infection accounted for 43% of community-acquired bacteremia, but only 10% of septic arthritis. Why is that? Well, Staph aureus produces surface adhesions that bind to cartilage matrix proteins. If you have staph strains defective in these adhesions, the rate of arthrogenicity is much lower, at least in a murine model. We think about septic arthritis as primarily being monoarticular and affecting large joints, particularly the knee. But septic arthritis may be polyarticular, affecting other organisms, and often associated with various reasons for immunosuppression and significant morbidity. In terms of the diagnosis of septic arthritis, this resolves to a considerable extent around synovial fluid analysis. We'd like to obtain gram stain and synovial fluid culture prior to the initiation of antibiotics therapy, but I'm sure all of us have had the experience of being asked to see patients who've already been started on antibiotic therapy. Synovial fluid analysis is useful. Synovial fluid white count helps us to stratify risk, although we know that many patients with septic arthritis have relatively low synovial fluid white counts. It's important to get blood cultures, both to assess the scope of infection, but also they may be the only source of a bacterial diagnosis. For the present, serum inflammatory markers are not, have limited diagnostic value. Since synovial fluid testing is somewhat insensitive, the rate of false negatives is in the range of 10 to 20% in many studies, our threshold for initiating antibiotic therapy should be low. And also, we need to consider broad-spectrum empiric antibiotic therapy. In the United States, we've seen in my career, my long career, the rise of methicillin resistance and a requirement for vancomycin. That's pretty routine for us. The problem is similar throughout the Americas and Asia, somewhat less so in Europe, but I suspect here as well, MRSA therapy is generally necessary. Gram-negative coverage is particularly indicated for critically ill patients and other patients with various reasons for immunosuppression. In addition to broad-spectrum coverage at the beginning of infection, it's important to narrow coverage as quickly as possible because of the morbidity of therapy over time. I was shocked in putting this lecture together how little objective information exists about what should be the duration of antibiotic therapy in patients with bacterial infectious arthritis. When I gave this talk to my rheumatology group at Yale, I realized that I was the product of my training, which is that we generally treat patients in our hospital for 6 weeks. But you'll find recommendations for shorter duration of therapy in the literature. I continue to recommend 6 weeks of therapy. One thing I think that's more clear, but not particularly on the basis of evidence-based studies, is the importance of joint drainage. In the pre-antibiotic area, patients would sometimes have good outcomes with aggressive joint irrigation alone. That's because joint irrigation decompresses the joint, improves blood flow, and removes bacterial toxins. We should continue arthrocentesis until resolution of effusion and culture negativity. And surgical drainage is indicated in a number of situations, such as hip infection, failure to respond to arthrocentesis, or soft tissue infections. So I mentioned that I wanted to give an update, and I wanted to focus on the idea of, has the landscape of infectious septic arthritis changed from the pre-biologic era to the present time? And there's actually relatively little data that I think we all bring our beliefs about this based on our own experience, but there's relatively little data to go on in the literature. Since I'm a guest of the European Union, I'm going to give data from 3 different countries. I'm going to start with the British registry data, which I think has the most information about this question of the risk of septic arthritis in rheumatoid arthritis patients when we compare biologic to non-biologic therapy. So this register, I suspect there are members of this in the audience, between 2001 and 2008 evaluated a significant cohort of UK patients who are being treated with etanercept, infliximab, and adalimumab, and compared their risk of infectious arthritis to patients on non-biologic DMARDs. What they found was that, and all these are obviously anti-TNF cohort patients, in the anti-TNF treated patients, the absolute risk of septic arthritis was fairly low, 4.2 per 1,000 patient years. But this risk was doubled in comparison to the patients treated with non-biologic therapy. There were 179 infections in the anti-TNF group compared to 20 in the non-biologic group. There was a signal of an increased risk of intracellular infections with organisms like Listeria and Salmonella. Interestingly, in this study, the greatest risk of septic arthritis seemed to occur at the onset of treatment. Was that because of a depletion of susceptible individuals in the exposed cohort? Could there actually be improvements in the patient's structure, let's say at the local level, that outweighed the systemic immunosuppressive risk of the treatment? That really wasn't addressed by the study, but it's interesting that in a Swedish registry, a similar observation was made. They evaluated about 4,000 patients who were hospitalized for infection, and as you can see, there was a time-dependent decrease in risk in the anti-TNF treated patients, so that by the 3rd year, their risk was pretty much back to normal. A final study I'll discuss is a French single-center retrospective cohort that evaluated patients seen in their institution between 1979 and 2002 versus patients seen between 2003 and 2013. I don't know that you can read all this, but if you could, you'd see that there's a lot of similarities. The main differences between the groups were that the biologic era patients were generally older, they had a higher risk for polyarticular infection, and not surprisingly, they had greater risk for MRSA. Overall, though, over the several decades that this study was evaluated, there was not an increased risk of septic arthritis in rheumatoid arthritis patients generally. So I think in terms of this question about the pre and biologic era, we can say that the absolute risk of septic arthritis in rheumatoid arthritis patients in the biologic, or at least the anti-TNF era, and other biologics I think have had similar results, remains low. But there is some increased risk compared to non-biologic DMARD patients. Most of this excess risk seems to come early in therapy, and there may be an increased risk of opportunistic infection and polyarticular infection. So as I've said, as I try to hit the highlights of changes in infectious arthritis, I decided to talk about Lyme disease and chikungunya, and I've already told you ways in which I think we should think about these things, these illnesses, perhaps having commonalities. So I'm going to begin by talking about the management of Lyme disease, and in particular Lyme arthritis, since this is a how to treat lecture. Lyme disease, Lyme borreliosis, is a spirochetal infection. There is borrelial heterogeneity, and the expression of this illness in North America and Europe and Asia are somewhat different. That's because in North America, Lyme disease is caused exclusively by Borrelia burgdorferi sensu strictu. Borrelia burgdorferi also causes Lyme disease in Europe in a minority of patients, but there probably are more arthritogenic strains of Borrelia burgdorferi in the northeastern United States, where I'm seeing patients as a rheumatologist, than there are in Europe and elsewhere. Other strains of Borrelia, Fsali and Guarini in Europe and Asia, cause skin and neurologic manifestations respectively, which are somewhat different than what I'm going to be talking about, but really aren't our subject for a rheumatology talk. Lyme disease is an emerging infection in North America, in Europe, and elsewhere. The disease has continued to increase in prevalence. In the United States, where data became available in the mid-1980s through the Centers for Disease Control, the reported cases of Lyme disease have pretty much increased each year since the mid-1980s to the present time. The reasons for this are not mysterious. There has been a change in the environment, a change in vector ecology, which has led to an increase in the hosts, deer, in the deer tick, as I'll show you, and therefore in the infection. I practice in Connecticut. As you can see, and I'm not sure how they determined this, it was determined that in 1900 there were 11 deer in Connecticut. This was considered to be a crisis. You may not know that in Connecticut we have a special relationship with Ohio. At one time that was the Western Reserve. General Cleveland was a Yale graduate. So perhaps that's why we turned to Ohio to get some deer. In spite of that, however, the rate of deer in our state was was relatively low until the 1940s. But since then, as a result of habitat modification, deer populations have increased exponentially in Connecticut. This same phenomenon is pretty much true throughout the temperate northern hemispheres. And as a result of this, Lyme disease has continued to expand. This shows you data from the United States, which is mirrored elsewhere. Basically, the increase in Lyme disease is not so much an increase of cases in endemic areas as geographic expansion of the disease. And that's been seen pretty much everywhere it's been examined. This is a worldwide problem in the distribution of Ixodes ricinus ticks. So let's talk now about the clinical features of Lyme borreliosis and get back to the concept of how we're treating patients with Lyme arthritis. Lyme disease is a bimodal infection, and we talk about early and late stage disease. Early disease begins locally as a local skin infection, erythema migrans. These patients have an infection in their skin, but they've not had hematogenous dissemination of the organisms, so they don't have constitutional symptoms to a significant extent. The typical sentinel rash of Lyme disease, erythema migrans, is shown here. This is a fairly large, circular, expanding lesion. The most common pattern is actually this homogeneous one. The bullseye pattern, which we think of as being more typical, is seen less frequently now because this pattern takes a while to develop. Physicians practicing in endemic areas are more likely to recognize erythema migrans before patients present to this bullseye pattern, and so you shouldn't look for this particularly. Early Lyme disease can progress from local to disseminated infection. For reasons that are not entirely understood, the organism disseminates to the nervous system in some patients, where it can cause meningitis or disease of the peripheral nervous system. It can disseminate to the heart, where it causes a fairly distinctive myocarditis. It can cause arthralgias and hepatitis. Dissemination may be more a feature of North American strains than European strains, and dissemination is less likely these days in endemic areas because the characteristic rash often leads to treatment that's curative. This just shows you an example of multiple disseminated skin lesions. In this rheumatology conference, let's get back to Lyme arthritis. I had an opportunity with Alan Steer a number of years ago to evaluate a cohort of Lyme disease patients who presented to our Lyme clinic at Yale with erythema migrans. This was in the 1980s, at a time when the cause of Lyme disease was not known. We thought it was a virus. So this cohort of patients did not receive antibiotic therapy for the treatment of erythema migrans. As you can see, 20% of these patients went on to develop no further manifestations of the disease. 20% of the patients went on to develop migratory arthralgias lasting for about 6 months. But more than half of the patients began to have intermittent attacks of arthritis lasting for days, months, or if these attacks lasted for longer than a year, we called this chronic arthritis. The pattern of involvement in the joints was very characteristic. As I mentioned in my introduction, when you think about Lyme arthritis, think about a specific clinical pattern, which is asymmetric oligoarthritis occurring in attacks, large joints, particularly the knee, often a lot of swelling, not as much pain in the joint as you might expect to see in other inflammatory rheumatic diseases. Small joint involvement is unusual. Symmetry is unusual. In these patients, none had more than 5 joints involved. As you can imagine, I practice in Connecticut. I see many patients who come to me and say, I've been to the rheumatologist, I've just been diagnosed as having rheumatoid arthritis. I don't want rheumatoid arthritis, I want Lyme arthritis. And in that situation, it's helpful to have a sense of what the clinical pattern looks like. And we'll come, let's move on now to treatment. Antibiotic therapy for early Lyme disease is oral. Doxycycline and amoxicillin are equally efficacious in Lyme disease. Doxycycline is more efficacious if there's co-infection with anaplasmosis. The duration of therapy is 10 to 21 days, and this is curative in more than 95% of individuals. I'm now going to switch and talk about the treatment of Lyme arthritis. Many patients with Lyme arthritis can be cured with a 28-day course of oral antibiotic therapy. If that doesn't work, do it again. and you'll capture more patients with another course of therapy. In patients who don't respond to oral antibiotic therapy, parenteral antibiotic therapy, usually with ceftriaxone, will resolve the infection and the arthritis for a significant percentage of patients. But what do you do if antibiotics don't work? And so I'd now like to talk about the problem of antibiotic refractory Lyme arthritis, which is seen in North America and seen in Europe and elsewhere also, but to a lesser extent because of different genotypic strains of the organism. So why are patients with antibiotic refractory Lyme arthritis not responding to antibiotic therapy? Is there a persistence of the organism? Is there some post-infectious inflammatory process? Here's a study from Steer's group looking at PCR in synovial fluid in patients with Lyme arthritis. This is a cohort of 88 patients, but not all patients were tested in each situation. 94% of patients with Lyme arthritis were PCR positive in their synovial fluid prior to the onset of antibiotic therapy. After a course of therapy, 65% of the patients became PCR negative. Only about a third remained PCR positive. But among the patients in this cohort who had persistent chronic arthritis, 7 of 10 were PCR negative. So the organism seems to have been cleared, but the arthritis persists. More recently, Steer's group has looked at microRNA expression in Lyme arthritis. You can see that the microRNA pattern in pre-antibiotic Lyme arthritis reflects an infection-driven immune response. But following antibiotic therapy in these patients with persistent arthritis, we now see a microRNA pattern which looks more like a chronic inflammatory immune response, similar to what's seen in rheumatoid arthritis. So it's not surprising that we would begin to think about using the same strategies in treating antibiotic refractory Lyme arthritis as we use in autoimmune inflammatory rheumatic diseases such as rheumatoid arthritis. What should we do in these patients? Historically, in an early ceftriaxone trial, it was suggested that corticosteroids might actually delay the resolution of Lyme arthritis, and I, for instance, avoided it for some time. More recently, pediatricians, European investigators have suggested that there's a role for simply doing intra-articular corticosteroid injections in these patients with antibiotic refractory arthritis. Here's a German study in a cohort of 31 children. 75% of the patients had resolution of their arthritis with 1 or 2 courses of antibiotic therapy. Then 8 of the patients received corticosteroid injections. In 4, the disease resolved, but 4 relapsed. With these recommendations and similar papers, I have started to do some corticosteroid injections, particularly in children, and frankly, I've been disappointed. Currently, I'm treating an 11-year-old girl. She came to me from the orthopedic surgeon. She'd received a corticosteroid injection after antibiotic therapy for her Lyme arthritis. The benefit lasted for about a month. She was then injected a second time. It lasted a few weeks. I injected her a third time and really got very little benefit. Now I'll get ahead of myself. I treated her with ceftriaxone. Within 3 days, her absolute neutrophil count was less than 500. So I switched her to methotrexate. She's been on methotrexate for about 3 months, and she's improving. So we'll talk about anti-rheumatic therapy. Initially, Steer and others, including myself, used hydroxychloroquine, but I think the evidence for efficacy of hydroxychloroquine is not there. There's very little published data about the use of methotrexate or biologics such as anti-TNF therapy in antibiotic refractory Lyme arthritis, but my own experience is that this works in many patients. We also looked a number of years ago at the use of arthroscopic synovectomy for antibiotic refractory Lyme arthritis in the knee. This is operator dependent and somewhat invasive, but it does work. We treated 20 patients with 22 knees because 2 of them were bilateral, and you can see that 15 of the knees responded both in terms of inflammation and a functional index, both immediately postoperatively and over 2-year follow-up. In 3 of our patients, there was inflammation improvement, but no functional improvement. We probably waited too long to operate on these patients. And in 4 of our patients, inflammation persisted in spite of this procedure. So my suggestions for the management of Lyme arthritis are that a majority of the patients can be treated successfully with 1 or more, perhaps 2 courses sometimes, of oral antibiotic therapy, amoxicillin or doxycycline. In patients who fail to respond, ceftriaxone will be helpful. Consider intra-articular corticosteroid injection. Consider a short course of disease-modifying therapy, because, for example, when we use methotrexate, we're able to discontinue this within a few months after patients go into remission. Consider also arthroscopic synovectomy, and consider or remember the natural history of Lyme arthritis. In the original study that I showed you from the 1980s, where patients did not receive antibiotic therapy, Lyme arthritis tended to resolve over a period of several years. Not that we want to have our patients wait a long time, but simply as you're making management decisions, it's helpful to have this perspective. So let me move on and talk about chronic chikungunya arthritis. Chikungunya is also a re-emerging infection. Chikungunya has been endemic in West Africa for centuries. Chikungunya was probably also found in the New World as long ago as colonial times. The virus is probably about 1,000 years old. For a long time, there was confusion, and chikungunya was probably being diagnosed as dengue. This changed in 1952 because of an outbreak of a crippling arthritis in Tanzania. The following year, the virus causing this arthritis was isolated from Aedes mosquitoes and recognized as a new alpha virus. The modern era of chikungunya begins in 2004, when there was an explosive epidemic of chikungunya in Kenya, with human to mosquito, back to human transmission. And since then, there have been worldwide epidemics of chikungunya affecting millions of people. There are 2 vectors of chikungunya, both Aedes mosquitoes. Aedes aegypti, the yellow fever mosquito, is also the primary vector of dengue and now Zika virus. This is an African mosquito. It's transmitted throughout the world, often on container ships. If you think about retreaded tires containing standing water, you couldn't find a better way to move these mosquitoes around the world. This mosquito feeds primarily on humans, and it's tropical. You can see in tan where chikungunya was in Africa in the 1980s and how it spread particularly to Asia and the New World since then. The other vector is Aedes albopictus. This vector became more important after the 2004 reunion outbreak because of a mutation in the outer envelope of the virus. This mosquito feeds opportunistically. It has both artificial and larval habitats, and it's more temperate than Aedes aegypti is, and it's displacing it in some locations. You can see a similar spread of this mosquito from Asia in the 1980s in green throughout the tropical and subtropical world. In the United States, it's important to me that we have Aedes albopictus because the range of these mosquitoes brings Aedes albopictus pretty much throughout the northeastern United States. In the Americas, most of the chikungunya that we see is from travelers returning from Asia and the Caribbean and South America, but there are locally acquired cases in Florida and in Texas. So this shows you the extent of chikungunya at the present time, pretty much throughout the tropical and subtropical world. Like Lyme disease, chikungunya occurs in two phases. There is initially an acute febrile illness, followed in some but not all patients by arthritis. Asymptomatic infection can occur, but typically following an incubation period of about a week, patients develop high fever, severe joint pain, and a maculopapular rash. This phase of the illness lasts for about 2 weeks and may be accompanied in patients with more severe disease, with gastrointestinal manifestations, with myocarditis, and in some patients with neurologic and ocular manifestations. Hemorrhage is possible, but less so than in dengue, although co-infection is certainly possible because it's the same mosquito vectors. Following this acute phase, up to 50% of patients will develop arthritis. This is often painful, disabling, and chronic. It occurs, as I'll show you from some studies, in a polyarticular pattern that particularly affects hands and feet, and it may be a rheumatoid arthritis mimic in terms of the clinical phenotype. Often these patients have severe pain and disability, and in the presence of large-scale epidemics in the developing world, these epidemics of disability have a significant economic impact. I've had an opportunity over the past several years to work with a colleague, Jose Kennedy Amaral, in Brazil. Chikungunya arrived in the Americas in 2013 in St. Martin and in the Caribbean. Since then, chikungunya has spread throughout the Americas. There have been more than 3 million cases reported since 2013. Chikungunya arrived in Brazil in 2014. My colleague Kennedy Amaral practices in Pernambuco along the equator, where there are tens of thousands of cases in his state alone each year. Recently, we evaluated a cohort of 50 patients retrospectively that he had treated for chronic chikungunya arthritis. We defined chronic chikungunya arthritis as a disease duration of at least 3 months following the onset of chikungunya fever. In our patients, the mean time between the onset of chikungunya fever and their presentation to the rheumatologist was 14.2 months. That's typical of other studies. Patients with chikungunya arthritis often are living with the arthritic symptoms for months, and we as rheumatologists are seeing them. We are seeing the most severely affected cases who've had chronic disease. In our cohort of 50 patients, there were 46 women and 4 men, and that's been seen in some, but a female predominance has been seen in some but not all studies. 60% of our patients had arthralgias only. 20% of our patients had both arthralgias and arthritis. All of the patients with arthritis had hand involvement. The pattern was polyarticular in about 2/3 and oligoarticular in the rest. We also looked at a separate cohort of 35 patients at disability measured by HAC, and we found the rate of disability in our cohort of chronic chikungunya arthritis patients was similar to rheumatoid arthritis. Another study that has looked at this idea that chikungunya in some ways mimics rheumatoid arthritis in terms of what it looks like is a study from Miner and colleagues at Washington University. They evaluated 10 American missionaries who returned from Haiti after traveling there in 2014, all of whom developed chikungunya fever. 8 of 10 of these individuals developed a symmetric polyarthritis that met ACR Eular criteria for rheumatoid arthritis. You can see the distribution of their joints in A. You can see the maculopapular rash that is characteristic of this infection in B. So how about the treatment of chikungunya? The treatment of the acute phase of chikungunya fever is supportive. No antivirals have been found to be efficacious. Some patients, particularly the elderly, the immunocompromised, need to be hospitalized for this illness. The mortality rate in chikungunya epidemics is relatively low, but there is some excess mortality in some series. Let's turn now to the treatment of chronic chikungunya arthritis. And I'm going to suggest again that there's a parallel to the treatment of antibiotic refractory arthritis, and that we'll need to think in these patients about treating what may be a post-infectious inflammatory process. So in terms of chikungunya treatment, NSAIDs provide modest relief. You have to be careful about possible co-infection with dengue and a risk for hemorrhage. Corticosteroids work, but are only really useful temporarily. Antimalarials, like Lyme disease, I think the evidence suggests are not effective. The best evidence for efficacy is for methotrexate. We recently did a systematic review looking at 131 reports of methotrexate in chikungunya arthritis and found that all of these reports were limited. 6 provided information that several of which I'm going to share with you. In an outbreak that occurred in Martinique in 2013 to 2015, there were 170,000 cases of chikungunya fever. The island's only rheumatology practice saw 128 patients with chronic chikungunya arthritis, and again you can imagine that these are the sickest of the sick, if you will, in a certain way. And as rheumatologists, this is an emerging area that we should be aware of, because we will see these kinds of patients. This report simply stated that 58 patients received methotrexate, 13 received anti-TNF therapy. They described the therapy as being effective, and perhaps more importantly, they found no safety signals in this report. The best discussion of efficacy in chronic chikungunya arthritis comes from an Indian study. This cohort evaluated 72 patients who were randomized either to receiving hydroxychloroquine monotherapy or triple therapy, as we're used to it in rheumatoid arthritis, with methotrexate, sulfathalazine, and hydroxychloroquine. As you can see, the triple therapy group had improvement in DAS28-ESR and HAQ score compared to the monotherapy group. But there are no randomized controlled trials of methotrexate monotherapy in chronic chikungunya arthritis. So, is methotrexate safe in chronic chikungunya arthritis? And this obviously comes back to what I was talking about a minute ago. Does chikungunya arthritis result from a persistence of viral infection? Or again, is this a post-infectious inflammatory process? There's not much evidence of persistent viral infection available in the literature. Here's a primate study in which in situ hybridization assays showed chikungunya RNA in mononuclear cells, and endothelial cells, and a few synovial cells in synovial biopsies in primates. In a study done in the La Réunion outbreak, only 1 of 49 patients with chronic chikungunya arthritis who underwent a synovial biopsy was found to have chikungunya virus staining in synovial biopsies. In contrast to this very limited information of persistent viral infection, there's a recent study by Chang and colleagues from Colombia, which evaluated 38 patients with chronic chikungunya arthritis by viral culture, quantitative reverse transcriptase PCR, and mass spectrometry, and virus was not identified in any of these specimens. So in the cohort that I presented earlier of our 50 patients, we treated 48 patients with chronic chikungunya arthritis with methotrexate and assessed their response in terms of pain with a visual analog score. Our patients' pain scores dropped from about 8 down to about 4 within 4 weeks, and we used quite low doses of methotrexate to achieve this improvement. So, chronic chikungunya arthritis occurs in about 50% of patients who develop chikungunya fever. In many of these patients, the arthritis is painful, chronic, and disabling. Methotrexate may be safe and effective, but randomized clinical trials are needed to assess methotrexate safety and efficacy. And with that, I'll thank you for your attention, and perhaps there's time for some questions. We're actually right at on time if we've been.
Just ask who's.
Any questions?
Yes. Thank you, sir, for your excellent presentation. I'd like to ask, Chikungunya arthritis, this is a sort of infective arthritis due to viral infection, or is it just a hypersensitivity reaction to Chikungunya virus or the mosquito bite, because they are treated by immunosuppressive?
Well, I think that we certainly haven't excluded the possibility of a persistent virus. But there's quite a bit of data in terms of the cytokine profile, that kind of thing, in which the signature in the synovium in chikungunya arthritis has parallels to what we see in other inflammatory rheumatic diseases. So I don't think we know the answer to your question, but I think we need more information in terms of treatment specifically about safety for that reason, and I think we need to better understand the pathogenesis of the disease before we can really answer that question.
Yes, thank you, sir, because viral infection may trigger the immune system itself.
Absolutely.
Thank you.
Yes.
Can you comment a little bit about the antibody testing in Lyme with Western blot or EIA, their specificity or sensitivity, if at all? And my second question is antibody refractory Lyme equivalent to reactive arthritis from Lyme, or is it a different entity?
Well, I think that let's start with the second question first. I think we can use somewhat different terms for the same things. I think we don't know for sure what the mechanism of antibiotic refractory Lyme arthritis is, but I believe that it is a post-infectious inflammatory process. How the term reactive fits in that context, I think, is less clear. In terms of the question about antibody testing, first, there's an issue in an international conference about whether we're testing for Borrelia burgdorferi or testing for a wider group of organisms. But let's focus on testing for patients who may have Borrelia burgdorferi sensu strictu. The biggest problem with antibody testing is not lack of specificity. It's the abuse of testing that occurs, particularly in the United States, something I'm familiar with, in which the predictive value of the test is diminished because the prevalence of the problem in the population is so low. And this leads to a real problem with false positive testing. And the second problem with false positive testing is misinterpretation of IgM results. So the IgM testing for Lyme disease is valid for the first 4 weeks of the illness, but often IgM testing is used to justify a diagnosis of longer-standing Lyme disease, which is inaccurate. Hopefully that answers your question.
Thank you.
So, hi.
Thank you. I'm a pediatric rheumatologist, and I would raise a question regarding duration of antibiotic treatment in septic arthritis. There is a pediatric study from Finland, a randomized controlled study comparing 10 days to 30 days. And they found excellent efficacy of 10 days with a quite rapid shift from intravenous to oral, but with a rather high dose oral.
I wish you'd been at my Yale Rheumatology grand rounds that I gave a couple of months ago, because originally in the slide I said 3 to 6 weeks. Yeah. And all my colleagues said, no, you have to treat for 6 weeks. So I didn't have the courage to have a 3-week part of the slide, and I think I tried to say that the answer to this question is not clear.
And then I would challenge the adult community to do a similar RCT and see if 10 days really gives the same efficacy and saves time and intravenous.
And the problem is, you certainly want to resolve the infection as completely as you can, but we're also dealing with the morbidity of therapy, and particularly in children with the need for ongoing parenteral therapy. So hopefully your study that you refer to and many more studies will give us a more clear answer about this, because I think many adult rheumatologists don't have that perspective. At least that's been my experience, but thank you.
So I have 2 questions basically. One, for how long methotrexate should be continued in patient with chronic chikungunya virus arthritis? What is the total duration? For how long the methotrexate should be continued? And my second question is that I belongs to Pakistan, which is an Asian country. We have seen a lot of patients with chikungunya arthritis, which later on we got the investigation done similar to rheumatoid arthritis, and they have positive anti-CCP. So is there any relation of this chikungunya virus with rheumatoid? Either this virus may cause to error in the genes and leading to rheumatoid arthritis?
Those are wonderful questions, and I don't want to keep anybody from lunch, but I'm very interested in both those topics. In terms of the duration of methotrexate in patients whose chikungunya arthritis resolves, there's no literature to guide us. In thinking about a randomized clinical trial, we're thinking about continuing methotrexate for 12 weeks after patients achieve DOS remission, but I don't think there's a clear answer. In terms of the rheumatoid arthritis parallel, I left a slide out for the sake of time. We reported a patient who had chikungunya fever from Pernambuco, Brazil, chikungunya titer positive, who went on to develop rheumatoid factor positive, CCP antibody positive, rheumatoid arthritis phenotypically, and Felty syndrome within a year after the onset of chikungunya fever. So that's just one example. As you point out, in a number of series, rheumatoid factor positivity and CCP antibody positivity is a measurable minority of patients. I personally believe chikungunya does not cause rheumatoid arthritis. I believe that the sensitivity of CCP antibody testing may be somewhat impaired in these large-scale epidemics. But I don't think we know the answer. And with that, I think everyone needs to go get lunch. Thank you for your attention.